Bismillahirrahmanirrahim. Assalamu alaikum. My name is Fatma Sleem, and today's topic is structure and significance of nucleic acid. Key concepts are for nucleic acid structure related to the structure of nucleic acids are obviously nucleotide di and triphosphate, structure of DNA, RNA structure and its type, and significance of DNA and RNA. We have previously discussed about the component of the nucleic acids and about the composition of the nucleic acids. So in this lecture we will discuss about the structure of nucleic acids. We have discussed in previous lectures that when a ribose or two deoxyribose are combined with the purine and or pyrimidine bases, they form nucleosides. So in nucleosides, no phosphate group is attached, and when phosphate group is attached to these two components, they form nucleotide. So basically here we have a ribosomal nucleosides, ribose nucleosides we can say them. These are adenosine, guanosine, cytidine and uridine. So nucleosides containing ribose are called ribonucleosides. The second type of nucleosides that contain a base which is attached to the deoxyribose sugar, and the nucleosides are called deoxyribonucleosides, and they have four types, which are deoxyadenosine, which is named as adenosine attached to the deoxyribose sugar, deoxyguanosine, deoxycytidine and deoxythymidine respectively. So you can see the structures of all these bases here, and they are present, these nucleosides are present in DNA structures and they contribute in the formation of DNA structure. Next is nucleotide. How nucleotide is formed? It is very easy. Base and sugar is called nucleoside, and when a phosphate group is attached to the pentose sugar, it will form a nucleotide. So when here in the slide you can see, it is very clear that ester bond is formed between the carbon number 5 of pentose sugar and phosphate group. So with one of the OH group on the sugar residue of a nucleoside to form a phosphate monoester. So when phosphate group attached to a carbon of one pentose sugar to form one nucleotide, it form a single bond with sugar and called monoester. But when it connects with two different nucleotides at the same time and make a chain, so one phosphate group is basically attached to the two different pentose rings of two different nucleotides. It will make a phosphodiester bond, which we have discussed in the previous lecture. So with the one nucleotide it is a monoester, and with the two nucleotides it will be the diester bond. In nucleotide, the sugar and the phosphate residue are connected by a phosphate ester bond, and in nucleotide the sugar and the organic base residue are connected by a N-glycosidic bond. So here we discuss phosphoester bond about the phosphate and the sugar, but we never have discussed about the base and the sugar, and this bond is called glycosidic bond. So what is basically glycosidic bond? It is a bond between sugar and sugar carbon 1 and carbon number 9 of base. This bond is also called nitrogen carbon linkage, And for purines, it is present between the carbon number 1 of pentose sugar and carbon number 9, sorry, nitrogen number 9 at purine, in purines. But in pyrimidines it is present in nitrogen at one position and carbon of one at one position in the sugar. So this is a basically covalent chemical bond, and it is stronger bond obviously. After nucleoside discussion, we are going to discuss about where the phosphate ion attached. So there might be two possibilities. It can be attached at the position 5 and it can be attached at the position 3 prime carbon of pentose sugar. There are two possibilities, and they are attached when they form a chain. So the sugar residue of nucleoside to form a 3 prime nucleotide. When it will attach to the 3 prime carbon of the pentose sugar, the nucleotide termed as a 3 prime nucleotide, and when it is attached to the 5 prime carbon on the sugar residue, the nucleotide is to form a 5 prime nucleotide. So we can say that in nature 5 prime nucleotide are more common. So they are dominant on the 3 prime carbon and sugar nucleotides. So we have discussed about the nucleotide. When a nucleoside is attached to the phosphate group, this is termed as nucleotide. And it could be possible to add 2 or 3 phosphates group to a same nucleoside, so that these nucleotides are very useful. And here we can see in the picture that base nucleosides, and then we add a phosphate group which make it a nucleotide, and we called it mononucleotide. Then there is another phosphate group which is attached to the previous phosphate group and make it dinucleotide, diphosphate nucleotide, nucleoside diphosphate. And then the third phosphate group is attached to the second phosphate group and make it nucleoside triphosphate. So why it is important to make a nucleoside triphosphate? Because these nucleotides carry chemical energy in the cell. Nucleotides may have 1 or 2 or 3 phosphate groups covalently linked at the 5-hydroxyl of ribose sugar. So these nucleoside triphosphates are used as a source of chemical energy to drive wide variety of biochemical reactions. So when these bonds between two phosphate groups are broken or hydrolyzed, they release large amount of energy, and which is used for the different function in the performing in the cell. Next, again the same thing, adding a phosphate to a nucleoside monophosphate generate a nucleoside diphosphate, and example of this is adenosine 5 diphosphate, 5 prime diphosphate ADP, which is what we call. And the picture is really clear. Adenine base and ribose sugar make a nucleoside adenosine, and with the one phosphate it is called monophosphate AMP, and with the two phosphates group it is called adenosine diphosphate, which is termed as ADP, and with the 3 phosphate groups it is termed as adenosine triphosphate ATP, which is more high energy bonds, and when break. So adding a phosphate to a nucleoside diphosphate generate a nucleoside triphosphate. Very simple. We have discussed in previous two slides, and we have see their picture as well, that clearly show how it is added and how these energy bonds are very rich source of energy. So nucleoside triphosphates such as adenosine 5 prime triphosphate, which is also used as ATP and used in almost every cycle where energy is required, are immediate source of energy for most of the energy requiring processes that take place in living things. So this is very important, adenosine 5 prime triphosphate. So this is also a important thing to discuss over here, that adding phosphates to a nucleotide is also important. Besides from the DNA structure, these are energy source as well. Then we discuss about the cyclic nucleotides. Cyclonucleotides are formed when one phosphate group has two phosphoester connections to the same nucleosides, monosaccharides. So we have discussed before, or didn't discuss it before. We only discussed the chain of nucleotides. Chain of nucleotides is in which nucleotides are connected in the form of chain, and phosphate groups connects these two nucleotides. So here this is different. Here phosphate groups is attached to 5 and 3 positions of the same pentose sugar and make a cyclic nucleotide. Polynucleotides. Nucleotides can be connected to one another with one another to form oligonucleotides, 2 to 10 nucleotides residue. But this is, however, not true, because we will discuss in the next lectures that how oligonucleotides are ranging from 100 to 50 to 100. We can synthesize them by ourselves, and greater than 10 is polynucleotides. So we will discuss them in the next lecture. More than 10 nucleotide residues is termed as polynucleotide, and the polynucleotides are mostly polymers called DNA and deoxyribonucleotides, and RNA is made up of polymers of ribonucleotides. There is no much difference between both of them. Some differences will be discussed in the same lecture today. So, first of all, structure of DNA is going to be discussed over here, that every cell nucleus in a particular living organism contain the exact same DNA molecules. So there is a one species, and every cell of the species contains same DNA in their molecules. Human DNA contains 3 million paired deoxyribonucleotides residue and contains 25,000 genes. So this is a very big amount. Genes are stretches of DNA that carry the code of protein production, code for the protein production. So the structure of DNA is understood in terms of three levels of structure. DNA, which are primary, secondary, and tertiary. So we will discuss them individually in the upcoming slides. First of all, we are going to discuss about the primary structure of the DNA molecule. Here we can see in the picture that how nucleotides are add up to each other. Um, the primary structure of a nucleic acid refers to the sequence of its nucleotide residue. First, a single strand is formed by adding the nucleotide with the help of linkage, phosphoester linkage. The sequence of bases is read in a 5' to 3' direction. So we can see that guanine, 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 and then thymine, then guanine, adenine, adenine. So this sequence is read from 5' to 3' direction. Phosphate and sugar backbone is present outer side and bases are inner side, carry all information for the protein synthesis. So we will discuss in next slides that how these are information is encoded to the protein synthesis. The DNA secondary structure is the set of interactions between bases. Which part of strands are bound to each other? We can say that in DNA double helix, the two strands of DNA are held together by hydrogen bonds. The nucleotide on one strand base pair with the nucleotides on the other strand. So these two strands are antiparallel to each other and have complementary bases that are opposite in direction and make hydrogen bond with each other. So we can say that the DNA strands are antiparallel in antiparallel arrangement, with one running in 5' to 3' and the other running 3' to 5'. The Watson-Crick base pairing of the two strands largely determines the secondary structure of the DNA. All naturally occurring DNA are double-stranded for at least some of their lifetime. The two DNA strands are held to one another by base pairing. Hydrogen bonding between the bases attached to the sugar phosphate backbone. The base pairing is complementary. Complementary means that adenine always form hydrogen bonds with thymine, and guanine always form hydrogen bonds with the cytosine. And these are two different biopolymers strands of DNA that are joined together. Double-stranded DNA is fairly uniform structure, and the need for a regular structure is one way in which changes in DNA, genetic mutations, can be detected. So We can say that the structure of the DNA double strand is very uniform, very regular, and if there is a change in it, we termed as a genetic mutation. The fact that the adenine and thymine base pair and guanine and cytosine base pair are very similar in size, it means no gaps, no spaces in between the double helix. If there is some irregular places in the double helix, it means that something is wrong with the structure, and this signals the need for DNA repair system to fix the damage. So we also have our DNA repair system uh, in secondary structure to make it uniform and regular. Tertiary structure. The DNA double helix may be arranged in space and tertiary arrangement of the strand. So in primary structure, only linear nucleotide strand is formed. In secondary structure, it form double stranded DNA molecule, and in tertiary structure, it wind around each other. And how can you explain it that tertiary structure is 3D structure of the DNA molecule? The two strands of DNA wind around each other in a covalently closed circular DNA. This means that the two strands can't be separated easily. Because the DNA strand can't be separated, the total number of turns in a given molecule of a closed circular DNA is a constant. So this is a new information about the link DNA winding, that the total number of turns in a given molecule of closed circular DNA is a constant, that is called linkage number, or we can denote them with the LK. So it remains constant for a given DNA molecule. For nucleic acid, tertiary structures refer to overall three-dimensional shape. In DNA, tertiary structures arises from supercoiling, which involves double helix being twisted into tighter, more compact shape. So we can see that plasmids, DNA from bacteria, these are the winded form of the DNA, and it is present at it is in the cytoplasm. RNA structure is just similar to DNA. Both are sugar phosphate polymers and both have nitrogen containing bases attached, but there are three main differences between them. RNA contains a different monosaccharide residue, as we know that in RNA ribose sugar is present, and in DNA deoxyribose sugar is present. So second is RNA contains the bases adenine, guanine, cytosine, and uracil instead of adenine, guanine, cytosine, and thymine. Third is RNA exists as a single strand instead of a double strand. So DNA molecules is the double strand molecules, and RNA molecules is the single strand molecules. Next is the significance of DNA and RNA. Obviously, we all know that DNA is a storage house of the cell, and RNA is used for the protein synthesis. DNA and RNA perform different function in human. DNA is responsible for storing and transferring genetic information, while RNA directly codes for amino acids and act as a messenger between DNA and ribosomes to make protein. Together, they keep track of heredity information in a cell so that the cell can maintain itself, grow, create offsprings, and perform any specialized function it's meant to do. The types of RNA we are going to discuss here are the following three types of RNA make it possible for the encoded information carried by the DNA to be put to use in the synthesis of protein. So how the DNA contain information and how we can encode it into synthesize the protein. So first of all, we are going to discuss here transfer RNA. You can see the structure of transfer RNA in the picture. Transfer RNA are the smallest of the three types. It contains 73 to 93 nucleotides residue, and they carry the correct amino acids to the site of the protein synthesis. Next is messenger RNA. Messenger RNA are of variable size depending on the protein to be manufactured and contain the information that specific which protein should be made. This message is a sequence of RNA nucleotides that is complementary to the template standard of DNA. So if first of all RNA make a copy from the DNA which is complementary, and then the messenger DNA is not of a same size, or we can't say that it is a shorter or it is a longer. It is depend upon the which type of protein we are going to synthesize from. अच्छा जी, next is ribosomal RNA are the relatively long RNA strand, hundreds or thousands of nucleotide residue that combine with the protein to form ribosomes, the multi-subunit complexes in which protein synthesis take place. So it is variable. It is a longer RNA as compared to other two types of the RNA, and it combine with the protein to form ribosomes. So we can say that the ribosomal RNA is present in the ribosomes and is exported to the cytoplasm to help translate the information in messenger RNA into protein.